and I have had a lot of fun talking to students and to faculty, some of them old friends. So thank you. So I'm going to talk today about some work that our lab has done off and on for about 20 years, and then more recently in the last 4 or 5 years, somewhat more intensively. And that has to do with how astrocytes or non-neuronal cells in the cerebral cortex of the brain function alongside neurons and are actually intricate partners with neurons in circuitry that processes information or mediates learning. That's a one-sentence summary of what I'm going to tell you today. All right. So astrocytes were first described by the German anatomist Virchow about 170 years ago. And he introduced astrocytes as neuroglia matrix in which the nervous system elements are embedded. And then with the development of the Golgi stain, it was Golgi who actually first saw astrocytes. And this is one of his earliest papers. And he shows astrocytes as contacting the vasculature. And indeed, that remains one of the most important functions of astrocytes. So astrocytes are star-like cells. They are a subtype of glial, non-neuronal cells, which include microglia and oligodendrocytes. They are found in the entire brain and are as numerous as neurons in mammalian brains. And today, of course, we have markers for astrocytes, glial fibrillary acidic protein, GFAP, or aquaporin, lining the end feet that line blood vessels and forming the blood-brain barrier or allowing water to go through, but not other molecules. And initially considered as passive structural support, astrocytes have now been shown to be crucial for the development, plasticity, and function of neuronal circuits. People who do cell culture have known for 50 years or more that you cannot make neurons have synapses in culture unless you add astrocyte or astrocyte medium. And a lot of work, including at McGill by Keith Murray and others, has shown the importance of astrocytes for development, for plasticity, for disease. Today, my goal is to talk to you about function in adult brains, particularly in the adult mouse cortex, of astrocytes working with neurons to shape circuit activity and behavior. Okay, so astrocytes are structurally heterogeneous. I thought I'll show you a picture from a wonderful review by Keith, where Keith points out that there are so many different kinds of astrocytes in so many different brain regions. Mueller glia, amazing astrocyte-like cells that span the entire retinal depth, or in the cerebellum, Bergman glia. Our work focuses on cortical astrocytes, and cortical astrocytes contact I'm sorry, I'm going to move this around here and there just to make sure you can read my titles. So cortical astrocytes tile the cerebral cortex and its layers from the PL surface down to layer 6. And here is a picture from 2 of my postdoctoral fellows, Grayson and Jennifer. Both have moved on to their own, Grayson to his own lab, Jennifer to a company. Green shows astrocytes with their filamentous structure expressing GCAMP6X, which is a sensor, which is a glutamate sensor that we use for multiphoton imaging. And red, shown in red, are a bunch of astrocytes expressing a construct called Chrome-Q, which is an optogenetic tool for modifying glutamate uptake. And one of the most amazing, certainly impressive things about astrocytes is, of course, their morphology. You can see this at low power on this side, the filamentous, umbrella-like morphology, which penetrates or invaginates synapses. Here is a picture from this paper of PSD95, which marks the postsynaptic density. And there are literally thousands of PSD95 puncta under this one astrocyte. So we know that a single astrocyte can contact tens of thousands of synapses. There's a paper that says it's 100,000 synapses. I think that's a little bit of a stretch, but at least 10,000, I would say. I don't know what Keith thinks, but thousands of neurons, tens of thousands of synapses. It tells you immediately that astrocytes have mechanisms for listening in on synapses, for modulating synapses, and not just one synapse, tens of thousands, large numbers of synapses. And immediately, let me plant the computational idea. Astrocytes carry out very high dimensional modulation of neuronal populations. Okay, if there's one thing you remember from my talk, remember that computationally, in terms of adult function, because astrocytes are present all over in the adult brain, that at least as numerous as neurons. Some people have said there are several times as numerous as neurons in the human brain. That too may be a little bit of a stretch, but each astrocyte remarkably can impact thousands of neurons, which in the field of information processing and circuit physiology means it has a little bit of effect on lots of neurons. Hence, processing that involves large numbers of neurons are likely to be affected. That is a theme of my presentation today. Okay, so astrocytes regulate neuronal activity via many different mechanisms. Astrocytes do not generate action potentials like neurons, so they are invisible to the microelectrode. That is why it has taken us so long to understand what they do. You need optical methods or methods to measure astrocyte calcium, which is the currency of astrocyte information processing and conveyance. Astrocytes generate calcium signals, which calcium is released from intracellular stores in astrocytes. Why? If they sense synaptic activity via glutamate and GABA transporters, shown in this very complicated, I apologize, schematic, when a presynaptic terminal releases transmitter, if it's excitatory, glutamate, if it's inhibitory, GABA, and then there are transporters, active proteins that suck up the transmitter that is not bound to the postsynaptic side. And if they do this actively, you can imagine they are shaping the spatial and the temporal elements of synaptic transmission. Astrocytes also have receptors for neuromodulators. In the cerebral cortex, a very important receptor is that for norepinephrine, which is released by norepinephrine locus coeruleus terminals and can activate astrocytes in order to lead to this red haze, which is the depiction of calcium signaling. In turn, and astrocytes in the striatum have dopamine receptors. Astrocytes release neuromodulators, most notably ATP, which is the stuff of energy which gets converted to adenosine, which can modulate synapses via adenosine receptors, particularly on presynaptic elements. And as I've told you, a single astrocyte can influence the activity of thousands of synapses and neurons. And a critical driver of our knowledge of astrocyte physiology is multiphoton imaging of calcium activity. And you can see here, this is just an image from work in our lab where neurons are labeled in red because there's a red sensor called JR Gecko is driven by synapsin promoter, and astrocytes are labeled in green, which is GCAMP driven by the GFAP promoter, and one can simultaneously measure neurons and astrocytes in animals that are doing interesting things, and you can ask, how do they work together to represent and process information and create action, which is the stuff of neuroscience. Okay. Astrocytes also, and this is where I have this famous picture from Cajal in human, a fetal brain of astrocytes lining blood vessels. I put almost under in brackets. I personally believe all astrocytes actually contact blood vessels. They have two ends to them, so to speak. On the one side, They are listening in on synapses, modulating synapses. But they also are contacting blood vessels. They are a crucial intermediary, therefore, between neurons and synapses and the vasculature. And indeed, by labeling astrocytes with a green GCAMP marker and the vasculature with this molecule dextrin Texas red, put in through the tail vein, which finds its way into the brain, you can see the close apposition between astrocytes, particularly the end feet of astrocytes, and the vasculature. So with this preamble, I'm going to tell you 3 short stories. The critical driver of our thinking is something that you might find obvious, but it bears repeating. The structure of astrocytes and the molecules they express are critical to their function. Okay, so I've given you a flavor of their structure and some molecules that they express, and I'll tell you how astrocytes sense neuronal activity to regulate blood flow and vascular dynamics, the size and volume of blood flow, which is critical for brain function. Astrocytes regulate the availability of glutamate and GABA at synapses to modulate neuronal information processing and learning. I'll tell you our experiments that demonstrate that. And I'll tell you about how astrocytes are activated by neuromodulators, in particular, norepinephrine in the cortex, and the signaling to astrocytes critically is instrumental, surprisingly, for learned behavior, in particular reinforcement learning. Okay, so first, how do astrocytes sense neuronal activity to regulate blood flow? So I'm going to take you to experiments that we did many years ago, and I was delighted to be talking with Ed and David about the ferret visual cortex a short while ago. We are all recovering nicely, but I shouldn't say that. Ferrets are wonderful animals for studies of visual cortex, but particularly visual development, because they are born very early in development. You can have access at postnatal stages to brain states, brain development that you have to go prenatally for most other animals. The experiments I'm going to tell you today have nothing to do with development. They have everything to do with a feature of the visual cortex that involves the representation of orientation. So there is a technique called optical imaging of intrinsic signals, which actually measures the delivery of oxygen via oxygenated hemoglobin, and the oxygen finds its way out of the vasculature and is used by neurons for energy consumption. And they need energy because they are responding to stimuli. Hence, if you take a ferret and you take pictures of the cortex at work simply under long wavelength light, you can get images like this, where you show one orientation, in this case, horizontal orientations, and with minimal processing, you can see these dark spots. And these dark spots are where oxygen has been given up by oxyhemoglobin and converted to deoxyhemoglobin, hence it absorbs red light. And these overlie neuronal groups that are responsive to horizontal orientations. Similarly for oblique or vertical. You take all these images and you do a pixel by pixel vector summation, and you get orientation maps like this, which tell you at a glance the representation of visual orientation in the primary visual cortex of ferrets. There are these columnar structures shown in green and yellow and blue, and that meet at pinwheels. So orientation maps with pinwheels or singularities arise from self-organizing rules of map development. And that's where, by altering development, we could show that the sizes, but not the nature of pinwheels, changes or can change. What does this have to do with astrocytes? Okay. So first, astrocytes can be labeled, and to two postdoctoral fellows in my lab many years ago, James Schumers and Hongbo Yu, did that by labeling astrocytes with this molecule called sulforhodamine 101. You just pour sulforhodamine, by pour I mean dribble, sulforhodamine onto the cortex, and you label neurons with Oregon green BAPTA, which predated GCAMP and other fluorescent sensors. And so you can see here green neurons and white astrocytes because they are expressing both Oregon green and sulforhodamine. And what you see here is that when this cortex is now being imaged under 2-photon microscopy at single cell resolution in response to different orientations shown on the bottom right, there are regions of this cortex, which is about 300 microns or 250 microns by 250 microns, in which both green neurons and white astrocytes are co-active. Okay, so for obliques, it is over here. For other obliques or for verticals, it's over here and so forth. Astrocytes in one region, like neurons in one region of this image, respond to one orientation. That's the point. Therefore, they are selective to this one orientation maximally. Hence, this is a map of orientation selective neurons and astrocytes where there is a pinwheel in the center because groups of neurons and astrocytes with different orientation preferences go around the clock in this one image. And so visual responses of neurons and astrocytes shown at a pinwheel demonstrate that astrocytes share precise response properties with neurons. This sets the stage for asking, do they have a role in the intrinsic signal blood flow image that I showed you? And to answer this, we had a hypothesis that astrocytes, actually we knew from, again, work of Keith. You know, he wrote a paper. I was telling him about this briefly in 2006. I urge you to go read it. The Journal of Neuroscience, Haber et al. I remember this paper because it changed my view of astrocytes. And very soon after that, we decided to work on astrocytes and derived the inspiration for this work, which in 2 years led to this paper in Science. And this paper, Keith showed that when you have cultured neurons and astrocytes, astrocytes have processes that are right opposed to synapses. Okay, these were slice cultures, not just two-dimensional cultures, they were slice cultures. And this argued strongly, and that the processes were dynamic, just as the synaptic processes, pre and postsynaptic, particularly postsynaptic processes would move. These would move too. So you could tell at a glance, they must have something to do with each other. And then there were these ideas that astrocytes have different sets of molecules that could be effective at synapses, such as glutamate transporters, which can suck up glutamate. So James and Hongbo did this beautiful experiment where they filled a pipette with red-colored, artificially, a molecule called TBOA, which is a blocker of glutamate transporters. And they put it in and they imaged this ferret under a 2-photon microscope. And what you see here is that before TBOA, in the control condition, you can see astrocytes in these white spots, in these white circles, and neurons in the green arrows. But then you iontophorease TBOA, hence you are blocking the glutamate transporters on the astrocytes. And the astrocytes go silent, meaning that their calcium signaling is critically due to their uptaking glutamate. And then post-TBOA, their response recovers. So it tells you astrocytes have calcium responses when they can soak up glutamate at synapses. There is a driver for this. We did not know what the driver was. Even today, it is not completely clear. How come glutamate uptake releases calcium from intercellular stores, though we have better ideas? And so that is what is shown here. Here is the pre Here is the during TBOA response, 0, and here is the post TBOA response back for astrocytes. Now, what does this do to the blood flow? Blocking astrocyte glutamate transporters kills blood flow related changes in the cortex. And that's what you see here, that in the region where TBOA is infused, the normal response to, in this case, two stimuli overlaid on top of each other is called the global signal, or the response to one stimulus subtracted from the other. It's called the mapping signal. This is what shows up in high contrast, those blobs that represent orientation-specific cells. It's completely wiped out when you block astrocyte glutamate transporters. What does this tell you? So here is before and after TBOA, the magnitudes of the response delivered by oxygenated blood and its conversion to deoxygenated blood. It tells you that the blood flow that brings the oxygenated blood has been blocked when you block astrocytes. And hence, astrocytes are responsible for a key component of hemodynamic brain imaging signals. And I remind you that when astrocytes are blocked, when astrocyte glutamate transporters are blocked by TBOA, neurons are responding away. In fact, neurons are responding more because there is more glutamate floating around. And you see that here. There is more neuronal response. Yet, the signal for oxygenated blood is wiped out. Therefore, astrocytes are a critical intermediary between neurons and the vasculature for driving blood volume signals. I can tell you in more detail as to what the various signals are. There is blood volume, there is actual deoxygenation, etc., that contributes to the fMRI signal. But the blood volume signal is a critical signal. And then we have gone on more recently to actually measure the calcium in astrocyte end feet and relate it to the vascular diameter. And what you see here is that as the green astrocyte end feet goes up and down in terms of its calcium signal, the diameter of the blood vessel shown in red goes up and down. So there's this close opposition between the astrocyte end feet calcium and the blood vessel diameter. And I also wanted to mention that the difference between the end feet and the blood vessel diameter is called the glymphatic space, and that's been prominently mentioned recently in the context of removing stuff from the brain or bringing in stuff that are larger molecules than what will go through the blood-brain barrier. Okay, so, sorry. There is more complexity in the relationship between astrocyte calcium signaling and the vasculature. This review, many other reviews have pointed out, and work has pointed out, that astrocytes release a bunch of different molecules. A great many of them, when astrocytes take up glutamate, a great many of these molecules, including potassium or prostaglandins or nitric oxide, lead to dilation of the vasculature. But there are also molecules like arachidonic acid that may lead to contraction of the vasculature. And we are studying this in detail. My own view is that it depends on the resting state. If a blood vessel is compressed, it has a lot of upside potential. If a blood vessel is already dilated, it has very little upside potential. It can only go down. There is a homeostasis to the regulation of blood flow. And resolving that is important for the field, even to understand how fMRI works even. But in general, there is no question astrocyte calcium increases in end feet lead to increased blood flow. Okay, so that's one part of my story. Astrocytes in visual cortex have tuned calcium responses to visual stimuli in ferrets. But in mice, it's been more difficult though even there because mice have salt and pepper arrangement. But if you look at say, the whisker region of mice, you do see very clear tuned calcium responses. So there's no question that these responses that astrocytes have are driven by synaptic astrocyte glutamate uptake by glutamate transporters. And this is the reason why they can regulate local activity-dependent blood flow by speaking to the vasculature and making it expand. And reducing astrocyte calcium responses by blocking glutamate transporters nearly abolishes the blood volume component, the dilation component of hemodynamic brain imaging signals. Okay? All right. So then next, how do astrocytes, by their regulation of glutamate and GABA at synapses, modulate neural information processing and learning? All right. So astrocytes have transporters. Transporters are a very critical component of astrocyte function. I told you about glutamate transporters. I'll tell you more, but let me tell you about GABA transporters. Here's an inhibitory synapse. And this inhibitory synapse releases vesicles of GABA. And some of that GABA that is not used is taken up by GAT1, which is the neuronal GABA transporter. But particularly around the synaptic space, in this what is called the extrasynaptic space, sit astrocytes with GAT3 GABA transporters. So astrocytes express GABA transporter GAT3 in the cortex, and this is responsible for reuptake of extrasynaptic GABA and maintaining the ambient GABA level. Maintaining the ambient GABA level is a crucial thing for maintaining not only a functional EI drive at networks, but also for even things like the resting membrane potential of neurons. Okay? And we wanted to ask, how do astrocyte GABA transporters shape visual information processing in neurons? One of the other reasons why the study of astrocytes has been complicated or certainly delayed with respect to neurons is not only that it's been difficult to study their functional properties till the advent of fluorescent imaging technologies, and particularly single cell resolution imaging via multiphoton microscopy, but also there have been lack of tools for discretely, specifically manipulating astrocytes. The GAT3 gene is quite complex, but Jin Tang, working with Rudolf Janisch, my colleague, and with Grayson in my lab, developed the strategy of AAV construct, of an AAV construct packaging into it multiple CRISPR guide RNAs, which are then targeted to different regions of the gene and is delivered into transgenic mice expressing Cas9 to delete GAT3 in astrocytes reliably and specifically. And then the idea is, if we deliver it into the cortex, along with neuronal imaging sensors, such as red JR Gecko, can we then examine the effect of deleting GAT3 from astrocytes on visual neurons? So that's what you see here. This is not a hole in the brain physically. This is a GAT3 deleted region in which there are neurons that express red calcium sensor. So this allows us to image these neuronal responses within and outside the GAT3 deleted region. And here are two basic ideas. What we don't find is any change in the tuning of a single neuron to orientation, let's say. Neurons within the GAT3 deleted region versus outside have the same level of single neuron tuning. Amazing. But then what we do find are changes in the population encoding. Here's one idea, that you can build what is called a generalized linear model, where you ask, how do different neurons contribute to the activity of one neuron? There are many other kinds of GLMs. I'll be telling you about them, because that's a way to examine population. Population coding. Hubel and Wiesel did the did the paradigmatic single cell coding study. You record from a single cell, you show it different orientations. I just showed you a little example of that. This cell has a tuning curve. This is how a single neuron encodes the world via this one property. It's a one-dimensional encoding, okay? In reality, I like to say this, and I apologize to Ed and David because they trained with Mike Stryker, who trained with Hubel and Wiesel. Hubel and Wiesel got lucky. Most encoding in the brain is not one-dimensional. You have many, many features that are represented, that are required to be represented in the activity of a neuron, which is a complex thing because the neuron gets so many different inputs from below, from the thalamus, from other neurons, from above, from other cortical areas. And all of this has to be represented in the activity of the neuron, in addition to not only visual stimuli, but internal state, arousal, attention, etc. So a way to analyze this is to build what is called the generalized linear model, where you take all these variables and you assign coefficients and you say, how can all of these variables explain the activity of the neuron? Okay, so here what Jiho Park, a very talented graduate student, did was to build a model that included just other neurons. And then she was able to show that the correlation between clusters of neurons, or the ability of a bunch of neurons to drive to explain the response of a single neuron, is highly reduced when you knock out GAT3. That's what this means. And that's due to increased noise and reduced coordination, not just correlation, because we're looking at many, many neurons. But the really critical demonstration is here. In addition to building models of how a bunch of parameters influence a single neuron, you can ask, how does a population of neurons represent a stimulus? By building a decoder, okay? And when you build decoders to drifting gratings, these gratings have 8 different orientations, or sometimes 16. Is the response to one orientation sufficient for us to read out what that orientation is? That's the basic question that a decoder answers. And you can take half your population and train it, and then you can ask for the other half of the population, how did it do? And when you ask this question of neurons in control brain regions or in control mice, they do pretty well. You can 90% of the time, you can tell what was the orientation that was shown to these neurons. But when you knock out GAT3, the decoding falls becomes much worse. And you can do this for gratings. You can do this for a set of 6 or 8 movies. Can the neurons decode a movie? So what does this tell you? It tells you that the decoding analysis of visual stimuli by drifting gratings of natural movies shows that this GAT3 knockout impairs neuronal population representations of stimulus information. I bring you back to the idea. Astrocytes speak to or influence large numbers of synapses, large numbers of neurons. And if you ask the question in the right way, you can see it. And so that's what is happening here, that when you block the GAT3 transporter, there is much more GABA floating around. And this GABA is utilized by at least these 3, if not more, kinds of inhibitory neurons. It's an intricate network. It is not a simple inhibitory network of one I inhibiting one E. And it is difficult to explain this on a cell-by-cell basis. But by recording from these neurons and doing population encoding analyses, you can decipher what astrocytes do. So astrocyte GABA transporters coordinate network activity critical for information processing. A second example of how does the glutamate transporter work. So here we asked, is there a role for glutamate transport in shaping synaptic plasticity that underlies learning? And here is the simplest learning task we could think of. And it was actually pioneered by Takaki Komiyama's lab. And I don't know if you can see, is that me? Sorry, I should have There is a sound to this, maybe, but okay, never mind. Here's a mouse. Here's a mouse. There is a lever in here. You have to, I hope you can see that. Okay, and this mouse is holding onto this lever with one hand, and the lever can go up and down or back and forth, and a tone comes on, and the mouse has to push a lever. Okay, it's the simplest of tasks. So it's a water-restricted mouse, head fixed, there are daily training sessions, and the lever push happens in response to a cue. And so they have to learn this sensory motor association. And they learn as they get better to have a smooth trajectory so that there is a component of the task that involves smoothness and trajectory. And so this is what happens in these mice. Between day 1 and 3 and 5 and 7, these mice become pretty good at this. That's the hit rate. And they learn to push it at faster and faster delays. Less and less delay. Tone comes on, they're pushing. Tone comes on, they're pushing, and they're getting a water reward. And between the early naive mice and expert, the trajectory becomes smoother because a push counts as a push once it crosses a threshold. So they become efficient at this. So now we altered astrocyte signaling in 2 ways. One, by genetically deleting glutamate, the GLT1, which is the critical glutamate transporter, via GLT1 knockdown in motor cortex astrocytes, and second, by modulating the astrocyte calcium via GQ dreads in motor cortex astrocytes. Astrocytes have this interesting property. You cannot inhibit calcium with a GI dread because of the way that the calcium release works. Not only us, others have shown this. You can only increase and clamp astrocyte calcium, which then blocks transients. And that is the way to study how astrocyte calcium regulates any function. Okay, hold that in your mind. And then we imaged neuronal activity to ask what happened in these mice compared to a normal mouse or a wild-type mouse. And this is what happens. That decreased GLT1 and disruption of calcium transients in motor cortex astrocytes alter task performance. So if you knock down GLT1 in these mice, you get decreased message, you get decreased protein. The animal continues to perform well. The association is not affected, but the trajectories are affected. From trial to trial, they are more wobbly in their trajectory because the encoding of the trajectory, which happens in motor cortex neurons, as I'll show you in a minute, is affected. So GLT1 knockdown alters movement trajectories without affecting the hit rate, which is what you see here. In contrast, the GQ dread, which you can see here, elevates but clamps astrocyte calcium so that the variations in the astrocyte calcium due to sucking up glutamate at synapses is gone. This affects both the hit rate and the movement trajectory, which are both worse in these GQ mice. And these GQ mice have increased GLT1 as opposed to the decreased GLT1. Okay, so in the GLT1 knockdown, there is reduced signal correlations. Neurons are less correlated with each other. In the GQ pathway activation, neurons have increased correlation. They are going up and down in sync much more. Astrocytes at a glance regulate neuronal population responses, but also we can decode the movement trajectory, and the decoding is very poor when you either knock down GLT1 or alter the astrocyte calcium. So astrocyte manipulation alter encoding of task parameters, and the generalized linear model is worse as well for many of these components. And this all goes along with the fact that when these animals learn, their astrocytes upregulate a bunch of genes consistent with their learning, including a bunch of transporter genes. These are not, the GLT1 and GAT3 are not the only transporters. sucking up stuff from the outside world is a critical function of astrocytes, and they all lead to calcium changes. And all of that is blocked when you block the glutamate transporter or you clamp the astrocyte calcium. GLT1 goes down in the GLT1 knockdown and goes up in the clamped calcium, but both of them, by affecting synapses, alters the ability of neuronal populations to encode the movement. That's the bottom line. So astrocyte manipulations alter encoding of task parameters and movement trajectories. So in this part, I have shown you that astrocyte GABA transporter GAT3 modulates population encoding. Again, the word population, because it's very difficult to find just single cells affected uniquely by astrocyte manipulation. It's the population view that is deeply affected, of the population circuitry. Hence, task-dependent, context-dependent of stimuli by visual cortex neurons. In motor cortex, astrocyte glutamate transporter, GLT1, and calcium-dependent signaling regulate neuronal population responses to shape movement trajectories and task encoding. Okay? And finally, I'm going to tell you about the last bit, which is that astrocytes are activated by neuromodulators, and in particular in the cortex by norepinephrine. And norepinephrine is crucial for a kind of learned behavior called reinforcement learning. Okay? Let me tell you first about reinforcement learning. Why is it interesting? Or why is it important? Reinforcement learning is the way we discover the structure of the world to inform decision making. Okay? So reinforcement learning is a trial and error process by which an agent, a child learning red means stop, green means go. You are not born with this information. You have to interact with the world. You may be told this by your parents, but you can figure it out by just looking at a bunch of traffic flowing, or you can figure it out even worse by, going into an intersection. Okay, so you act on the world, and the world tells you this is rewarding or not, and that updates your model of the world, and then you act on it again. If it's just a binary signal, green versus red, go versus stop, you just need one bit of information. But if it's a complex world, you need to interact with it many, 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 many times to understand its structure. And there are theories and there are models for how reinforcement learning, or this action reward contingency, should shape brain circuits or a computer program or an algorithm in order to understand the world. So to develop an optimal policy, an agent faces the exploration-exploitation dilemma. Exploitation means you want to maximize the current reward. That is a given for most agents, including human beings. You want to exploit a known action-reward contingency. But if you simply get stuck on one thing in a potential well, you will never explore other areas where there are more rewards to be had. So you also want to have a balance between exploitation and exploration. Continue searching the environment to find a potentially more rewarding outcome. And the goal of all this is to acquire an optimal policy efficiently. And to do that, you require measures of value, to measures of uncertainty, and of context. And let me tell you why this is interesting and important, particularly for understanding artificial systems, or AI. Deep reinforcement learning has enabled superhuman performance in challenging games and control tasks. For those of you who follow DeepMind, Google DeepMind. DeepMind used to exist before it got acquired by Google. DeepMind's big insight are in a set of papers in Nature and Science, where DeepMind showed, and this is Demis Hassabis and his collaborators, that by exploring and then exploiting, by assigning Q values to to a set of actions and then evaluating them, an algorithm can figure out by itself how to play the game of chess or to how to play the game of Go, and it played them so well that it beat the world's best Go player. And I remind you, Go has more possibilities than there are atoms in the known universe. So there is the space of moves is infinite almost. And the same with chess. By the way, for chess, those of you who play chess, in 1997, was it, that Deep Blue, this IBM computer, beat Garry Kasparov? Since then, algorithms have way outperformed humans, so much so that the latest chess championships take place between humans, but the championship between algorithms take place at a much higher level, at superhuman levels with much higher what are called ELO scores. And compared to the best algorithm, the DeepMind chess algorithm beat that in 99% of games. And how did it do it? It is by a reinforcement learning algorithm where you have a tree structure, a Markov tree structure, where you try certain things and you see and you evaluate it and you play against yourself millions and millions of times. Okay? So in games with Markov states and deep search, what is the Markov state? A Markov state is where there's no history. If I'm playing a game of chess and I walk away, I have to go to the bathroom, and I ask Yang to play in my place, Yang can walk in and play. He doesn't need to know how I got there. There is no history to the game. What you see is what you have in front of you. These are very well posed problems for reinforcement learning algorithmically. But solving real world problems with reinforcement learning algorithms remains elusive because we need to assign reward on extended timescales, on behavioral timescales. If you want to be admitted to the McGill Neuroscience program, you have to do well in your classes, have a high GPA, apply, get interviewed. When you get your acceptance letter, that is your reward. But the reward has to be ascribed to that whole chain of events, which by themselves are unrewarded. The reward only comes at the end. It's called the credit assignment problem. It is a deep problem for algorithms, but we do this all the time. Okay, almost nothing we do is immediately rewarded. So extended signaling on behavioral timescales for credit assignment or for transfer learning. If you learn something, can you apply it to something else? We do it all the time. Yet for algorithms, there is catastrophic forgetting in these kinds of situations. So new insights and computational framework derived from biological reinforcement learning are needed for next generation machine learning. Okay, so whether you're interested in next generation machine learning or not, just I want to put it out is that AI doesn't do everything better than human beings. Human beings do some things better, and is worth understanding why. So neuromodulation is key to reinforcement learning, and we have known this since the discovery of dopamine as a signal for reinforcement learning, going back to the work of Wolfram Schultz. But Wolfram Schultz's work had to be married with Rescola and Wagner's theories about reinforcement learning. So what did Schultz show? Schultz showed, recording from the ventral tegmental area, that you take a mouse, you take Pavlov's, not a mouse, a monkey. You take Pavlov's dog, for example, and you give it a reward. You give it meat, and the VTA will fire. Then you ring a bell, and you give it meat, and after a while, just the bell itself, as the conditioned stimulus, will make the neurons fire because the activity of dopamine has shifted in time. It's called temporal difference learning to the stimulus that predicts the reward. Okay, it's basic reinforcement learning. And then if you omit the reward, there is a decrease in firing of the VTA neurons, and that's a surprise. And the animals learn very well from that surprise that the game has changed. Okay, so surprise is a crucial learning signal. However, dopamine cannot be the only story behind reinforcement learning. Subpopulations of dopamine neurons in VTA encode many, 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 many things. I'm not going to get into it, but there's a whole slew of literature on this. And other neuromodulatory systems also show signatures of reward and prediction. And what are these systems? That is dopamine, of course, shown in red here, including the VTA, the ventral tegmental area. But there is norepinephrine shown here, and acetylcholine shown here, and serotonin. And the interesting and important thing that I like to point out is that these are all deep in the brain, physically and metaphorically. They are all these are all ancient brain systems. And that is because it is crucial for the brain. The brain has evolved to live in the world. If the world did not exist or didn't exist in this way, we will have a different brain. We need systems in our brain to understand the world we are in. Every organism and every organism's world is different. But every organism has these basic properties, these principles by which it needs to understand the world. And that's the point of neuromodulatory system, at least one point, okay? So then, dopamine and norepinephrine, another reason why dopamine cannot be the whole story is because of the anatomical distribution of dopamine. Dopamine terminals are heavily infiltrate the striatum, but in the cortex, they only go to the very front of the cortex, whereas norepinephrine goes everywhere in the cortex, but avoids the striatum. So they have complementary distributions, which leads us to ask, and there's a literature on norepinephrine before our work, but that literature implicates norepinephrine in gain modulation for movement. Norepinephrine bursts come before an animal in a simple task was asked to move, or of course with arousal. So the questions we have asked is, do locus coeruleus norepinephrine neurons, norepinephrine is made by this structure called the locus coeruleus. It sits just below the cerebellum in both mice and in humans. And in mice, there are about 3,000 neurons, astonishingly few neurons, but they spread, they have widespread projections, and they have deep impact. So do these locus coeruleus norepinephrine neurons have a role in reinforcement learning, and in particular, signaling this thing called reward prediction error? What is reward prediction error? It is the surprise. You are expecting a reward and reward does not happen. That is a massive error, and it leads to suppression of dopamine, and I'm going to show you massive release of norepinephrine. Or you give a reward when none is expected, and that too has the same effect. So do LCNE neurons signal this? And if they do, how is this used in the brain for interesting things, such as implementing reinforcement learning? Two questions. And I'm going to tell you that LCNE neurons do have a massive role in reinforcement learning, and importantly, astrocytes are crucial for implementing across time reinforcement learning in the brain. Okay? Okay, so here's our task. So here's a mouse, and this mouse is sitting here, and here's its front paw and is holding a lever. And we train this mouse. By we, I mean they, okay? When I say I mean we. And when I say we, I mean they. But it is so much fun for me to at least sit in on these experiments and at least not live my life totally vicariously by simply hearing about results when after they have happened. So that's just a little, this is what happens to older scientists, sadly. But anyway, so what did Vincent and Gabby Drummond do? They trained this mouse on two tones. There is a low frequency tone that says, do not move. And there is a high frequency tone that says, move. And move means push this lever, and if they do so correctly, meaning beyond the threshold, they get a water reward. And they have been mildly water deprived, okay? If they don't press to the go tone, that's a miss, and nothing happens. On the no-go tone, if they do press, that's a false alarm. Remember that. That's a crucial thing for us. It's a false alarm. The animal thought it should move. Maybe it heard a go tone when it was really a no-go tone. Or maybe it was in a different brain state. Maybe it was thinking about what am I going to do in my home cage when I go back there. whatever, it moves and it gets an air puff. It's a punishment, it's a mild punishment, okay? And if it doesn't move and it shouldn't move, that is a correct reject, and that's nothing happened. So these are the contingencies, okay? And these are the ways in which the mouse acts on the world of lever pushing and reward and updates its internal model to see, to match our experiment. And there is one critical variable in our experiment, which is the tone intensity can vary. And therefore, that adds uncertainty to the go or no-go stimuli. It can be very weak or it can be very high. When the go tone is very high, highly intense, loud, there is very little uncertainty. If the go tone is low or low intensity, there is much more uncertainty. Okay? So here's what the mouse does. Sorry about that. And that was the go tone, so the mouse moved. That's a no-go and the mouse does not move. And you get that here. Okay. So what happens? So these mice learn this task. As the go tone increases, they get better and better. They get more and more hits, as they should. On the no-go side, it is not a symmetric situation, but nonetheless, there is some mild relationship with the intensity of the go tone and reduced pressing, as there should be. But here's the really interesting result. When the mouse pushes, when it shouldn't, it's a false alarm and it gets punished. Then on the next trial, the animal's behavior is much better. The animal decision boundary has shifted so that on the next trial after a false alarm, there are fewer false alarms, there are more hits, and hence the D prime, which is the difference between hits and false alarms in units of standard error, goes up. Okay, and that's a standard way to demonstrate performance in behavior. Okay, so this is called a serial response bias. And this is a crucial driver of learning because something surprising happened. The animals are not at 100%. They still have upside, and they are still surprised because they push expecting a reward and they get air puffed. And so they immediately shift their decision boundary. This is a learning signal. Okay, and we know that it is so because we can put a laser down there and block the locus coeruleus by making it express this inhibitory opsin. And when we do that, then the current trial response, go tone, push. At the lowest go tones, performance drops, meaning that norepinephrine release, which I'm going to show you in a minute, is really critical for exploration. On the next trial, after a false alarm has happened, and the false alarm only happens in 10 or 15% of trials because these animals are pretty well trained. But when it does happen, on the next trial, the number of hits goes up, the D prime goes up, but if you block the locus coeruleus, that increase in hits and the increase in D prime does not happen, meaning that the behavior does not get better when you block the locus coeruleus. Okay, that's the point of this. So suppressing norepinephrine release reduces execution on the current trial when evidence is weak. Norepinephrine promotes exploration, and suppressing norepinephrine release blocks improvement on the next trial after a false alarm, suggesting norepinephrine is a learning signal. And here are the neuronal responses from optotagged units in the locus coeruleus. What you see here is that pre-movement, there is a norepinephrine release. These are the activity of locus coeruleus norepinephrine neurons. And post-reward in a hit, the animal moved correctly, it got water reward, there's a little bit of norepinephrine. And therefore, when in the pre-press, the louder the sound, the more is the response of these neurons. In the post-hit, meaning reward given when expected, The louder the sound, the lower is the response, meaning that there is less and less error in my prediction. Hence, the response of norepinephrine is lower when the go tone is louder. It is a direct demonstration of reward prediction error, which has not been so easy to show, even in Wolfram Schultz's work, where he does not study reinforcement learning. You have to study reinforcement learning with variable stimulus reward contingencies to discover these. But after a false alarm, before the animal moves, the animal heard something. It thinks I should move. There is a little bit of norepinephrine. The animal moves, and then it gets an air puff. And the norepinephrine response goes through the roof because the animal was expecting a reward. It got punished. It is a massive reward prediction error. And you can see that these responses are the highest responses we have ever seen from locus coeruleus neurons. And you can see that at a glance here. So pre-movement responses correlate with expectation of reward. Post-reinforcement responses correlate with reward, uncertainty, or surprise, both for a hit and certainly for a false alarm because they actually signal reward prediction error. And so then, for the purposes of my talk today, how does this false alarm signal, which leads to improvement on the next trial work? It is a learning signal. And that finally brings us to astrocytes in a minute. So there are two ways by which the signal can be conveyed to the next trial, which is 6 seconds away. It is possible that neurons keep firing. That's, after all, how short-term memory is conveyed in many cases. I tell you a number, it is a 7-digit number. You have to remember it for 5 seconds till you put it in your phone. There is some evidence prefrontal neurons can just maintain their activity. But that does not happen here. There is no persistent activity after a false alarm, and you can see that at a glance for even beyond a couple of seconds after a false alarm in the locus coeruleus or the prefrontal cortex neuron. Now we are recording the prefrontal cortex, okay? We want to see what is the signature in neurons that might mediate this learned decision boundary shifting. And you look at single neurons in the prefrontal cortex, and there is no persistent activity beyond the false alarm-driven activity in these neurons. So and yet this information is present. Remember, this is a very high dimensional space. We are recording from hundreds of neurons, sometimes thousands. And in that neuronal population is the information that a false alarm has happened, and now the next stimulus comes, and if it is a go stimulus, I should push. If it is a no-go stimulus, I shouldn't push, and the animal's decision boundary is much better so that it makes much fewer mistakes. What does this mean? It means that the representation of the go tone and the separation from the no-go tone has been increased because the animal hears better, if you get my meaning, okay? How can we show that's happening? Yes, if you record from PFC populations, you can show that norepinephrine increases the discriminability of go and no-go stimuli because you can calculate the population activity in the stimulus subspace on trials following a false alarm. What do I mean? Here's the behavior. After a false alarm, then the next trial, there are fewer false alarms, more hits. Here is the neuronal responses from hundreds of prefrontal cortex neurons. How do you analyze this? Here's one simple way. You boil down this activity into one dimension. You can do this in 2 dimensions, principal component 1 and 2. You just do 1 dimension. It's called the coding dimension. And you ask, what is the difference in the representation of activity of the go tone versus the no-go tone? And it is very significant after a false alarm compared to non-false alarms or other situations. So therefore, norepinephrine increases the discriminability. Yet there is no signature in the neurons of sustained firing, which leads us to ask, how is false alarm information conveyed to the next trial on behavioral timescales, on these long timescales? And the hypothesis is that astrocytes are directly activated by norepinephrine, and they generate prolonged calcium responses, and they enable reinforcement learning by reconfiguring neuronal circuits. So norepinephrine release, which I've shown you is a massive release, acts on astrocytes, which we know have norepinephrine receptors, and this alters the activity of millions of synapses and tens of thousands of neurons in a high-dimensional way to regulate behavior. And that's how this information persists. And in the next 10 minutes, I'll show you some, not 5 minutes. Oh my God, I've been rambling. 5 minutes. Is that okay? I will tell you, I'll show you the evidence. Cortical astrocytes reliably respond to norepinephrine release. You optogenetically release norepinephrine, and there's massive response from cortical astrocytes. In prefrontal cortex, in these mice that have been trained on this task, we can show that astrocyte calcium responses are strongly driven by false alarm. So this is the astrocyte response after an FF that follows a false alarm. In contrast, the neuronal responses are much, much, much more restricted. These responses last at least 6 seconds and sometimes more. And so the response duration of astrocytes is several-fold longer than the responses of neurons. This is totally consistent with our NeuroPixels recordings, okay? So astrocytes convert a phasic norepinephrine signal, which lasts only for 100 to 200 milliseconds, that's spiking after a false alarm, to a calcium signal lasting for several seconds, okay? This is a critical idea. And so then blocking the false alarm mediated calcium signal using astrocyte dread, if you block it, then it blocks the behavioral improvement. So there is much less improvement in the D prime behaviorally if you block astrocyte calcium increases with GQ dread, just as I showed you in the previous set of experiments. You can even block astrocyte calcium by knocking out adrenergic receptor 1A on astrocytes using a floxed ADRA1A mouse. Okay, and indeed, this blocks the rise in astrocyte calcium after a false alarm, and it blocks the behavior. And what does this astrocyte calcium do? This astrocyte calcium, we think, releases ATP into the extracellular space, which is converted to adenosine. This ATP release can be measured using GRAD ATP, and indeed, there is ATP release before and particularly after a false alarm. And this ATP release can be increased after a false alarm and is blocked by prazosin. So I was delighted to see in Ed's lab similar experiments in zebrafish. So that's amazing that there are evolutionarily conserved ideas. And blocking the adenosine action on A1 receptors blocks the behavioral improvement. So I'm building for you parallel stories of molecules being released, being measured, and the effects on behavior. And the idea is that norepinephrine acts on adrenergic receptors to increase calcium, which releases ATP, and adenosine, which acts on A1 receptors to modulate neuronal activity, which affects behavior. And how does it modulate neuronal activity? We can do NeuroPixels recording. This is my last data slide. And to make a long story very short, in controlled conditions, the post-false alarm activity level in the population is high compared to post-hit or unreinforced trials. And importantly, the D prime between go and no-go stimuli is high in control conditions, but it vanishes when you block astrocyte calcium transients or the A1 adenosine receptors that this signaling pathway acts on. So what this says is that PFC neuronal responses are altered by norepinephrine astrocyte signaling. We are altering the separability of go and no-go responses, and we submit this is sufficient explanation for the behavioral decrement following astrocyte calcium, astrocyte dread, or the DPCPX blockade of the adenosine A1 receptor. So in summary of this last part, phasic norepinephrine release from the locus coeruleus after a surprising outcome, which is a large reward prediction error, facilitates behavioral optimization in a reinforcement learning task. Okay. The high LCNE activity following a false alarm alters the population discriminability of go and no-go stimuli on the next trial. How does that happen? Here's a plausible mechanism for which I hope I've shown you some evidence. Cortical astrocytes are directly activated by norepinephrine, which is released from the locus coeruleus after a false alarm. This leads to this large increase. Even though this is transient, it leads to a large, seconds large increase in astrocyte calcium. This is the way you bridge time that is critical for reinforcement learning. Astrocytes exhibit large sustained increases in calcium activity, thus extending prediction error signaling in time. And this norepinephrine-driven astrocyte calcium and astrocyte ADP adenosine signaling to neurons increases the discriminability of go and no-go stimuli. So this astrocyte calcium leads to ATP and adenosine signaling, which acts on neuronal synapses. In a slice, adenosine inhibits excitatory transmission. In vivo, there is a lot of stuff going on. There is excitatory transmission to excitatory neurons. There is excitatory transmission to inhibitory neurons. There is an intricate network that I gave you a glimpse of when I talked about GAT3. In this intricate network, adenosine has a more sophisticated effect. It is task dependent, and this effect is to increase the separability of go and no-go stimuli. And so norepinephrine signaling to astrocytes extends a learning signal in time and is instrumental, it is causal for implementing reinforcement learning in the brain. So if there's anything you remember from my talk, specific astrocyte mechanisms, GABA transporters, glutamate transporters, and norepinephrine activated calcium signaling have well-defined mechanistic impact on neuronal circuits that underlie information processing and learning. Thank you very much.
Thank you very much. That was really very clear. And
I want to acknowledge various people
sorry.
in red who are currently in my lab on the left, and those who used to be in my lab who contributed to the experiment that I've talked about.
Very important. And the credit assignment problem can never be ignored. All right. Are there any questions from the audience? Okay.
Beautiful talk. Thank you so much for all that information summarized in a very short amount of time. The question is about the third part. So there's a literature about the role of norepinephrine in attention. And I'm wondering if it could explain partly your result. That is, it's not that much a reward prediction signal, but it's just that the aircraft should, lose the attention of the animal in some ways.
You repeat the question.
Yes. So the question is that there is a literature that links norepinephrine to attention. And is it possible that the air puff alters attention or increases attention that is responsible for the improvement in behavior? And it is an outstanding question. And the answer is that we can never eliminate that possibility. I was talking with Dana Small today, and even in her experiments, reinforcement learning due to a reward prediction error, which is by definition a surprise, because it is And the larger the prediction error, the more surprising it is. In fact, there is a theory of attention actually being a prediction error about the world. So that's one kind of answer. But the second kind of answer is that we have done experiments. They are in our bioarchive paper, which we are, let's say, having a less than amiable conversation with a major journal about for the last 1 year that I actually mentioned to some of your students with whom I had a wonderful 1 hour. In that paper, which is on bioarchive, you will see a set of experiments in which we not only elicit reward prediction error with an airpuff, but on a correct reject trial, when there's a no-go stimulus, the animal does not move, but you give it a reward. So it is called a correct reject reward trial. So it's opposite valence. And you still get a massive norepinephrine signal. And you get a change on the next trial. But it is a different kind of change. In the case of a false alarm airpuff, you get an improvement in behavior on the next trial. In this case, you get a decrement in improvement. And you can imagine the mouse got rewarded for doing nothing. It heard a no-go tone, it didn't move, and there came a water reward. It thinks momentarily, the world has changed on me. Now I need to do nothing. And indeed, when a go tone comes, it does nothing. The misses go up. So it makes sense. The behavior makes sense, but the underlying mechanism still goes through norepinephrine and through astrocyte calcium, and it reorganizes neuronal activity, but in different ways. It's still a mystery as to how do the same astrocytes know what has happened in order to change, but we can't solve everything all at once. But a very good question.
Are there any students? Yes, Patrick.
Thank you very much for that amazing talk. A couple of years back, Nicolas Brunel published a paper about modeling working memory, because before there were modelings about working memory only with neurons. Now he proposed to introduce astrocytes. Do you suspect that your findings, especially with norepinephrine at the end, could be a way to explain astrocytes impact of the model?
It is possible, absolutely. If I remember the paper right, my students keep giving me papers to read, and I have a hard time keeping up with them. So if I remember it right, it did not invoke any particular mechanism, but invoked the idea that astrocytes are in a good position to integrate lots of information and convey slow analog signals. To that extent, the spirit of that paper is very much in line with what we are demonstrating.
A couple more questions.
Yeah, thanks so much. That's really fascinating. As you made a strong point that neurons, that astrocytes are regulating large groups of neurons, but in like the calcium video you showed, for example, you can see how there's different domains of the astrocyte that become active because of subdomains of calcium activity. However, do you think that there's like a functional organization to that? Do you think within a domain it's for a specific subtype of neuron, or ones that are like temporarily locked or anything?
Again, outstanding question. It is true that astrocytes are not homogeneous, just like a neuron. But in my opinion, the parallels between astrocytes and neurons are actually very high. Just like a neuron gets different sets of inputs onto different parts of its dendrites. And if you image a neuron's dendritic arbor, you will see activity as inputs come in till they sum to threshold, and then the soma fires. It's my image of astrocytes, based on quite a bit of data we have, is not dissimilar. That the currency of astrocyte activity is calcium. This calcium is released from intercellular stores, which are all over the place and through IP3R2 receptors. And when glutamate transporters, which are all over the place, or even GABA transporters, or certainly norepinephrine receptors in the striatum dopamine receptors, when they are activated, these are G protein coupled, they can lead to local release, which can sum up to lead to the soma activation, which is what we are measuring. But the activity in the arbor can tell a story of all the stuff that astrocytes respond to and can integrate. And someday it will. I don't know whether I will live to see it, but somebody will. You will. Yeah, but there will be a theory one day of astrocyte integration, just as Rawls' cable theory had a big impact on describing how neurons integrate. But this will involve calcium, calcium dynamics, calcium flow, and calcium summation.
One last question. Keith?
Beautiful presentation, Mriganka. Thank you so much. I have a question about heterogeneity of the astrocytes. And so in your talk, you covered multiple astrocyte populations. And I was wondering how much of this functional properties of these astrocytes you think comes from them being highly specialized and tuned for the system that they're in. So for example, if you took a visual cortex astrocyte and you put it in somatosensory cortex, do you think it would function in the same way and have the same functional properties?
That's another outstanding question. Even in the prefrontal cortex, again, in our yet to be published bioarchive paper. I know, this is like a, anyway, never mind. There is not that all astrocytes respond in the same way. We can cluster them based on their responses using formal clustering techniques. There are at least 2 or 3 different subsets of astrocytes. There are separately measured using RNA-scope. The ADRA1 receptor expression level varies from quite high to quite low. So we know that not all astrocytes are responding to this norepinephrine release, nor can they. So there is variability. What does it mean? I don't know the answer to that. It is very likely that the results we are describing is based on the 50 to 70% of astrocytes that do respond to norepinephrine with prolonged and large calcium increases. What are the other 30, 40% doing? I don't know, but they are not doing this. Maybe they're doing something else. Maybe there are specialized functional classes, even in PFC, even in prefrontal cortex. And certainly between visual and frontal cortex, there may well be, people have done RNA sequencing of astrocytes, and they do see different things. My friend Baljit Kak has done a lot of studies on the striatum, and they are definitely different from cortical astrocytes. At the very least, functionally, they have dopamine receptors, which cortical astrocytes have very little. So because dopamine is a big player in the striatum. So that tells you also that astrocytes are, quote unquote, tuned to their world or the structure that they are in.
So Bernice Grafstein actually has been tuning in through Zoom, and she just wanted to let you know that she's truly been enjoying your talk. I could see her laughing on the video with joy, so thank you very much. And so please join me and Bernice in thanking Mriganka Sur for their talk. So there'll be a little wine and cheese reception outside, so please join us, and you can continue conversing with Mriganka there. Let him have a little wine, though.
So can I just break out?
We can just finish.
Okay. Oh, I see. Dr. Grafstein, I wanted to thank you for endowing this lecture and for the opportunity to be here. All right. Thank you very much. Yeah.
Thanks, Bernice.
Say something. Okay, I'm signing off.